Thank you so much. I am beyond humbled to be included in this list of esteemed speakers, and I've been really, really interested in the talks that have happened today. So I'm hoping that my talk is up to par with the others. Today I'm going to talk about the work that we've been doing the past about 10 to 15 years in developing transcranial magnetic stimulation protocols for use in autism spectrum disorder. I am required to disclose that I am a federal government employee, so the views I'm expressing here today and over the weekend are not necessarily those of the federal government. But what they are influenced profoundly by pioneers in the field of autism. And one of those pioneers who I had the great privilege to meet was Dr. Isabel Rapin. And Dr. Rapin was one of the original people who identified that autism is a brain disorder. It's a disorder that is based in neurodevelopment. And when I met her, she was well into her 80s and was still doing research. She passed away just a few years ago. But as we all know, and we've been discussing, and many of you know quite well, it's a behaviorally defined disorder, and the behaviors per the DSM-5 are these two core deficits, and as has been discussed over the weekend, many associated symptoms. And when I started in this field, we were trying to identify the brain target, the brain mechanism that we thought was going to be the answer to identifying, diagnosing autism. And this was a figure that is now several years old, but explains some of the brain regions that were thought to be critical in autism. And as we moved on in the field, we realized that it's not just the individual regions, but rather how they're interacting and how they are forming into networks. And so, there we go. My PhD was actually on identifying the functioning of the mirror neuron system in autism, which here is the MNS. And at the same time, there were sort of competing theories looking at the theory of mind networks and the sensory processing networks and other networks that still, again, were going to be the end-all be-all now at a network level. And again, we've sort of advanced over the years to now accept that there's not going to be just one, right? And as I showed, the symptoms are just too complex to even think about having just one underlying mechanism. So after my PhD, I spent some time thinking of, okay, if there's going to be multiple brain regions and multiple networks that are affected, how can we intervene on these multiple networks? Is there something that we can use that is flexible enough to target lots of different networks that may be helpful in developing new treatments? One last thing before we get into what that is and what transcranial magnetic stimulation can do, is to say that the great Isabel Rapin didn't say it was a brain disorder. She said it was a neurodevelopmental disorder. It's a disorder in how the brain develops. And we know pretty well, there's been a lot of very basic research in how the brain develops and the trajectory from cellular migration in the brain to development of whole networks and functional systems like that underlying basic sensory processing, receptive language, higher cognition, and these different networks have different timescales in terms of how they develop over time, and one builds upon the other. So again, not to say that one of these may be the underlying problem, but rather if we can intervene at various levels from cellular and molecular to systems, early sensory systems, or even later systems, that we can affect various symptom domains. And specifically what seems the sort of going theory right now to be driving this neurodevelopmental disorder seems to be in the development an imbalance in this inhibition and excitation system. And those can be measured in the form of molecules like GABA and glutamate that we've heard of. It can also be measured using magnetic resonance spectroscopy, which is like it's a sequence within the MRI where you can measure levels of concentration of GABA and glutamate in an individual in a region of the brain. And through development, as the brain is developing, this imbalance may be leading to changes in how the brain is responding to different stimuli, and then how those brain regions are connecting up into networks through synaptic connections. And there's a lot of literature suggesting that the end result here is that we have a brain that is hyper-connected, a brain that has too many connections, that isn't forming integrated networks like what we typically might expect. So this mirror neuron system, for example, is present in individuals with ASD. It's just maybe not as integrated, not as able to be automatically engaged given a certain environment. So how can we facilitate this? How can we strengthen the networks that should be strengthened and reduce down maybe this over-connectivity and encourage pruning, maybe enhance GABAergic transmission, etc., using a tool that is a large system level non-invasive tool. And that is where TMS comes in. Sorry, oh, there it goes. But it's not simple, right? I feel like I've sort of, every time I give versions of this talk, I kind of scare people off of like, oh my gosh, we thought it was simple, it's not simple. But that's not to say that it shouldn't, it's impossible to start. And I'm hoping this plays, because it's a really fun video. Let's see. Shoot. All right, that's okay. What you would see if the video would play, sadly, doesn't seem to be working, is evolution of this brain and how it's gotten connected. I can send you the link, it's on YouTube. But the idea here is that it is a ever-growing, ever-morphing set of connections that we can target. Okay, so here's the complexity, right? We got an elephant here, but there's a pun, if you will, a joke. How do you eat an elephant? Anybody know the answer? One bite at a time, right? So one network at a time, one system, one brain region at a time. We're starting to probe using non-invasive brain stimulation, using TMS, to try to eat this elephant, to try to uncover and modulate these brain networks and see what they do for the various symptoms of ASD. So what is transcranial magnetic stimulation? It is a device that essentially looks like a paddle. It's about this big. Place it on top of the head. And inside this paddle, this plastic enclosed paddle, are copper windings. And attached to this plastic enclosed paddle is a very large electric capacitor. So we build up electricity, we send it through these copper windings, and due to a very basic physics law called Faraday's law, Maybe I should have been paying better attention in my high school physics class, but because as I tell my children, you never know when you're going to use this information. Faraday's law, when you send a very large electrical current through a wire, it induces a corresponding magnetic field. The magnetic field in this case is perpendicular to the direction of the electrical current, just going like this inside the paddle. Perpendicular happens to be, if you place the paddle on top of the head, directly into the brain. Okay? So magnetic field goes directly into the brain, and again, thank you Faraday, induces a corresponding electrical current inside an electrically charged battery, if you will. Electrically charged battery happens to be the brain. The brain is electrically charged. We send a magnetic field down through the skull into the brain. Electrical current inside the brain activates the neurons below. And I know it does this because if I put it over your primary motor cortex, your finger starts twitching. I've activated your brain region corresponding to the motor control of your finger. So we can do this, and we can stimulate. When we stimulate, by the way, it feels like somebody's flicking your head. You can feel it, I'm not gonna lie. There's definitely a sensory aspect to it, but it's not as painful as those who have, I myself have undergone a electrical stimulation. So electrical stimulation, like if you do a peripheral nerve test, hurts. Electrical stimulation hurts. Magnetic stimulation, again, you can feel it doesn't hurt as bad. So we do this, we pulse this magnetic field into the brain, and it's going to activate neurons within about a centimeter or two, depending on the coil that you use, centimeter or two cube of brain tissue that's sitting right at the top of your brain, the cortex. It's going to hit all the cells and all the neurons that happen to be in that region. So it's not going to hit just inhibitory neurons or just excitatory neurons, but depending on how you pulse it, you can preferentially engage different types of cells, and we know this as well. We also know that the amount of energy that we're putting into the brain is pretty high. So just to give you a sense, to compare it to other devices that might be on the market, we have here TDCS, Transcranial Direct Current Stimulation, or TES devices, Alpha Stim, if you're familiar with that. That's down here in terms of the amount of energy. We have something called low field magnetic stimulation. We have the Earth's magnetic field, just to give you a sense, right? So the TDCS is just slightly higher than that and actually lower than a refrigerator magnet, if you're curious. We have the MRI, which people know is very big a magnet. And we have TMS that's about the equivalent about a Tesla of magnetic field that's going into your brain. So it's pretty high and induces a pretty large electrical current, lower than what you would get from electroconvulsive therapy. And in that way, we're actually able to control the electrical current and not induce a seizure in most cases. The risk of seizure is approximately 1 in 100,000. It has happened, but it is very rare. Okay, so basic things that you should know about TMS. 1, if we pulse it once, we get a twitch, right, if we happen to be over the primary motor cortex. If you pulse it multiple times, think about doing the same thing over and over and over again. You induce plasticity. And you can change, for long term, the functioning of a given brain region. So when they do this over the primary motor cortex and you have the person, you do a single pulse and the person twitches and you measure it, and then you put in low frequency, 1 hertz or less. So I pulse the device for a period of hundreds to thousands of times, once per second. We then have a phenomenon that lasts from one session, maybe half an hour to an hour, of reduced excitability in that area. By pulsing it once per second, the brain relaxes down. It increases GABAergic transmission, and we have increased inhibition, and you get less activation, less excitability following 1 hertz stimulation. On average, there's always those paradoxical responders that, like myself, who takes Benadryl and is up all night. But in most cases, we have this reduction. Opposite, you have high frequency stimulation where you have trains of usually 5 to 10 to 20 hertz stimulation, and it's going kind of in trains of 2 to 3 seconds. So you usually turn it on for 2 to 3 seconds, it goes, and then you turn it off for 8 seconds, and then you turn it back on. And all of these patterns were actually generated from what we think we know about how the brain responds in different situations, and what happens when that does. So when we do high frequency stimulation, we see facilitation. We have an increase of excitability for, again, 30, 40 minutes after the session. Okay, from a single session. And we're not, oh, I have to point it at, sorry about that. The other protocol, another protocol that I'll mention later that is critical to understand, is a protocol called theta burst stimulation. And in theta burst stimulation, similar to the low and high frequency protocols, if we did it to motor cortex, which is where a lot of the basic research is done, because we have a nice clean finger twitch to say what are we doing here, and we again do a single pulse, finger twitches, and then we put in a pattern. Instead of now just 10 hertz or 1 hertz or whatever stimulation, we do it in bursts. So instead of just going for 2 seconds, we're going, and turns out that pattern is much more related to what the brain will naturally be doing when it's inducing plasticity, when it's developing new connections and or pruning them. So a continuous form of this stimulation, called continuous theta burst stimulation, takes 40 seconds. That's it. 40 seconds of these bursts coming at 5 hertz, so 3 pulses at 50 hertz, very quick, but the bursts are coming at 5 hertz. Ends up 40 seconds is 600 pulses you get in 40 seconds, and that's for the continuous. And then there's an intermittent form where you do 2 seconds of this bursting followed by 8 seconds off. And the original studies that developed this, which were back in 2005, found that when you pulse it in this continuous pattern, just like the 1 hertz or lower stimulation, you see a suppression in excitability. You see the brain getting dampened down in terms of its excitability. But if you do it in an intermittent pattern, you see a facilitation, a greater response following the stimulation. And at least in the literature, when I came into this field, they said that the effects last, again, 30 to 40 minutes from a single session. So my first question was, well, I know in autism, brains are wired up differently. These networks and systems aren't necessarily going to behave the way we think they're going to behave. So let's just see, on a basic level, what happens when we do the same protocol that we think we know what's going to happen to in an individual with ASD. So what we did was we gathered up some neurotypically developing controls, and we first showed that yes, we could replicate what Wang and colleagues were finding in our data set of typically developing individuals. And then we did it in individuals with ASD. And this is what we found. So just to put the 2 graphs on top of each other to bring home my point, the effects lasted about 2 to 3 times as long. A 40-second protocol lasted well over an hour on average in individuals with ASD, and the effects were larger. Okay, this is good actually, because hey, guess what? I can manipulate the system, and whatever I do is going to last longer in the brain. I just have to know this going into then what I'm going to do in the brain. Okay. In addition, what we found was that when we stimulated using the continuous theta burst stimulation, and then we looked at the magnetic resonance spectroscopy after, we saw an increase in GABA concentration. So this suppression, we think, may be driven by an increase in GABAergic concentration within that region. We also found that despite the increase in GABA, you might think, well, that might decrease connectivity, but actually no. We saw an increase in connectivity following these protocols. And what I think is going on is actually that there might be, in this reduced functional connectivity in ASD, an issue of too much noise. So as we bring up the GABA, we bring up the inhibitory control, we're actually getting higher functional connectivity scores in terms of how correlated, how related the activity of point A and point B are, because I'm getting the signal from point A to point B more efficiently. So then we thought, oh, okay, now we might be onto something, and let's see, you know, how it might apply to ASD. I should also say that there's many millions of patients that have received this repetitive transcranial magnetic stimulation for treatment-resistant major depressive disorder. It is FDA approved, has been for over a decade, and multiple devices have been approved, multiple protocols at this point have been approved, 30+ years of clinical and research experience establishing safety, establishing efficacy in a different condition. Also in adults with anxiety, OCD, they recently just got FDA approval for RTMS in those conditions. So there's no reason why RTMS needs to be specific to adults, or that it needs to be specific to depression. It is a brain stimulation, brain modulation device. And so we can apply it, and we can apply it safely in children all the way down to newborns. It's used to suppress status epilepticus in brand new newborn babies. It is thought to be safe and, again, effective in a number of conditions. But there's not as much research out there in children, and there's not as much research out there in individuals with ASD. So, and you know, children with ASD. And so, and there's only been a few large-scale randomized clinical trials in children for our TMS, and for what it's worth, so far, they've all been negative. So let's get to the results. The first set of studies that were published came out of a lab in South Carolina. Manny Casanova did a number of studies where he went with this theory of GABA deficiency and said, okay, if we have a GABA deficiency in ASD and we know that these protocols can increase the GABAergic concentration, let's see what we can do if we place it over the dorsolateral prefrontal cortex. Why dorsolateral prefrontal cortex? Well, it happens to be a hub. It is a part of the brain that's connected with pretty much every other part of the brain and is part of lots of different networks. So what he found across many studies is an improvement in repetitive behaviors and irritability. These studies were all relatively small scale, 20 to 30, mostly children, and waitlist controlled, but not randomized, and he's published quite a lot. So there's potential here, right? There's not been any randomized controlled trial on this, with this protocol or on this target. Then again, I came in with this idea of the mirror neuron dysfunction. I said, okay, let's see what we can do with the mirror neuron system. We randomized about 20 to 30 adolescents and adults in a number of studies, and had some mixed results. You know, some individuals had amazing results, and really we were like, yes, we hit the target, this is great. We're seeing some real behavioral benefits related to social interaction and empathy and facial expression recognition. And then others who didn't see anything at all. Recently, another group has published a different protocol, but interestingly, it's 5 hertz. Standard protocols, not bursts of 5 hertz, but rather 5 hertz, to the same region, and noted improvements in adaptive behavior and daily living skills and communication. Again, to the same target, approximately the same type of protocol, and again, randomized here. So there, again, hope, potential in this target. There's a group in Australia who has done a lot of work in this area. Pete Endicott and his colleagues conducted a randomized trial stimulating dorsal medial prefrontal cortex, again with a 5 Hertz protocol. And saw some sort of mixed results. Again, some people responding really great, other people not. They didn't hit their primary endpoint. They hit some secondary endpoints in that original trial. And a group in China, or sorry, Chinese in Taiwan recently published some other studies, again, to a theory of mind network node region, this time the posterior superior temporal sulcus here, and they noted some improvements in repetitive behaviors, but again, mixed results here. Some people did really great, other people had no effect. I'm really excited to see the end results of this trial. This is the first of its kind, multi-site, large-scale targeting based on MRI and using this intermittent theta burst stimulation to a theory of mind and looking at social relating. Pete came yesterday through Zoom and promised that their interim data should be out by spring, so maybe I'll come back next year and let you know the results of that trial. Okay. So what about other associated symptoms? So I highlight this one also because this study specifically targeted children with intellectual disability and targeted a symptom of sensory motor integration, something not necessarily considered core to ASD, but maybe a really challenging, impactful symptom domain, and saw some success in terms of applying 8 Hertz stimulation to the motor, premotor system. Okay. Executive functioning, another associated symptom, has been looked at across a number of trials here. But what I'm going to highlight here is, if you notice, smaller scale trials, randomized, positive, another one is open label, positive. These both used ITBS. And then for some reason, they switched to a different protocol. Same region, same brain region, but now we're stimulating in a different way. And both of these more recent studies have been negative. Note they're also much larger studies than the previous ones and randomized. So I'm not sure yet, mixed bag in terms of executive functioning. Another recent study that just came out was related to connectivity in the parietal cortex, and when they stimulated using high frequency 15 and 20 Hertz stimulation, they saw some improvement in the language and social subscales, but again, this is open label. And lastly, well, it's FDA approved. We got a huge amount of data in depression. So why don't we try it in depression in the context of ASD? Well, how does that respond? And in fact, it responds pretty well across 2 now open label trials. All right. Now, response to our TMS is not only based on the TMS parameters, the frequency or intensity. It's also how your brain responds, also based on what state your brain is when it's getting stimulated. So a couple related pieces of findings that are relevant. There is a large amount of variability across all indications. So when I said some people got better, some people got worse, this is not specific to autism. We see this in depression, we see this in other indications. And there's a lot of work being done in the field to try to figure out how can we manipulate the state of the brain so that we can optimize the effects of the stimulation. And one thing that's come out of this is that the power and phase of an ongoing EEG oscillation is actually relevant to then the responsiveness of the brain to that stimulation. And I was super excited to get sort of a shout out from Dr. Gaetanis, who I'm looking around, I don't see him right now, who, you know, in thinking about how we can integrate this idea of closed loop EEG, right? We're recording EEG and we can stimulate when the brain is at a certain state, when it's at a, based on power of a certain frequency band or based on a certain phase of an ongoing rhythm. We also know that when the brain is at a high theta power, there's greater antidepressant effects. Again, theta burst stimulation, it makes sense, right? We also know that when alpha power is low and beta power is high, in healthy volunteers, you got the signal through the network faster and better. So maybe we want to get people into a low alpha, high theta state and then stimulate. We also know that activation downstream in major depressive disorder is most active when we stimulate at the trough of the alpha. So again, a low power state of the alpha rhythm. So we're taking in all this information, and again, you talk about repurposing drugs, repurposing the knowledge that we can apply it into ASD. We also need to remember, let's not forget my original work, that ASD brains respond differently to these protocols. So we need to do these sort of initial tests to say, okay, what's going on after just a single session? Then we can say, yes, we're on the right track. And we do know, again, from depression literature, that the immediate acute response in mood predicts then the long-term benefit. And so what can we do in terms of immediate outcomes, MRIs, EEGs, tasks or tests immediately after a single session such that we can apply this long-term? And again, understanding that this brain state is critical. We have identified a couple different mediators and moderators of response. For example, in one study they found that individuals with higher IQ had a better response, less severe ASD symptoms and social communicative and social impairments, less ADD, but more severe adaptive functioning. When I read these first three, I was like, oh great, like we've cut out like a huge portion of our population here. But it seems like those with more severe adaptive functioning actually responded better to the executive functioning protocol. We also know that certain psychotropic medications that may be affecting these chemicals may impact also the effects. So we got to know about the person that we're stimulating. We got to get some pre-measures to then predict, okay, who is this best for? So to answer my question early, so does it work in ASD? Maybe. For some things and some people, but most of the literature is really small scale, less than 10, 20 people. It's open label. There could be placebo effects. We have some ways to go in terms of randomized trials. I think there is potential. This has been my career for the past over a decade, and I plan to continue. But we need larger scale randomized trials. We need to stratify and select our participants more carefully. We need to figure out where and how to stimulate and develop more smart trials and kind of listen to less of the hype of, you know, the people saying, well, gee, you know, TMS can cure autism, or there's a doctor out there who says that my nonverbal child can all of a sudden get up and speak after a single session. I've never seen it. I've never seen it. So understanding that there's a lot of research that goes into this, a lot of variability, and it's not ready for broad clinical dissemination yet. So what can we do now? Because I realize we need it now. Well, if you're a sponsor, you can support more good research. And I'm saying this not for me, because now I'm at the National Institutes of Health, I'm fully supported. But for others and for these multi-sites trials, it requires a lot of resources. Stakeholders, parents, individuals with ASD, be informed consumer of the research. participate in regulated research trials. These clinical trials, you can find them on clinicaltrials.gov. Find the trials, participate. We need participants to make the studies happen. And don't go to the clinics and spend tens of thousands of dollars on unsubstantiated promises. That's my plea. Researchers, we welcome you to the table. I think I had a picture of this at our roundtable on Friday. We need more people in the field. I, along with Pete in Australia and Manny in South Carolina, can't move the field fast enough. So we need more people. We need more researchers in other areas of TMS to come into the autism area and to understand and apply those that are out there doing TMS, how having ASD may interact. All right, and I think about 5 minutes over, but I thank you for your time, and I am available to answer any follow-up questions via email. I told somebody earlier this, NIH, the beauty of it is everybody's email. If you ever need to find somebody at NIH, first name dot last name at nih.gov. Every single person's email at NIH is that way. So this is mine, and I'm happy to answer any questions. Thank you.
Thank you, Dr. Oberman. Thank you. Do you have any questions here? We'll just take one question and then we'll move on to the next talk because it's almost lunchtime.
What do you think are the possibility of trying to personalize the treatment by, for example, doing a quantitative EEG, trying to look at their levels of alpha, beta, et cetera, and then trying to tailor the frequency?
Yeah, so the challenge with that is there has yet to be any study that is linked and change from pre to post in any of those frequency bands with any clinical response. And so I don't think that that's a valid way to go yet. I do think that there is